Good afternoon, everyone. Thank you for joining us today. And my name is Young Rock, and I'm an assistant professor in the Department of Family and Preventive Medicine. And I'm also a relatively new member of this EGDRC. So happy to join in this amazing group. And it is my great pleasure to introduce today's speaker, Dr. Wendy McKimpson. So Dr. McKimpson is an assistant professor in the Division of Digestive Disease at Emory School of Medicine. And Dr. McKimpson's journey in biomedical research began at the University of Michigan, where she completed her undergraduate degree before moving to New York City to pursue her doctoral training. And she earned the Ph.D. at the Albert Einstein College of Medicine and completed postdoctoral training at Columbia University, establishing herself as a rising investigator in the field of metabolic disease. Dr. McKimpson's lab has developed the expertise in dissecting and signaling mechanisms of endocrine cell function and dysfunction, and also intersection of the metabolism and gut biology. So today, Dr. McKimpson will discuss her research on treating diabetes by targeting hormone-producing cells in the two key organs, the pancreas and the gut. Through the understanding these mechanisms, her work explores new approaches and also potentially the treat diabetes. So please join me in welcoming Dr. Wendy McKimpson. And Dr. McKimpson, so over to you.
Okay, thank you for that kind introduction. So with that, I'm excited to share my work with you today. And I know I'll just dive right in. And I know I really don't, particularly to this group, need to give an introduction about diabetes. But here's just to set the stage that in addition, within the US right now, we have currently 38 million children and adults within the US that have diabetes. But this number, when we think about it in a larger context, we actually have more individuals, 97 million adults within the US that are actually on their way to developing diabetes or have prediabetes. So when you put these numbers together, nearly half of adults within the US have both diabetes or prediabetes. And importantly, when we think about it in just the context of prevalence and incidence, we know that both incidence of type 1 as well as type 2 are increasing, not just in the United States, but also worldwide and globally. So it's definitely important to understand what's happening with this disease. And importantly for me, it's also a personal conquest in the fact that this is my mom on a recent trip. Well, recent, this was now more 5 years ago, but she's had type 1 diabetes for over 65 years. So I've personally seen just the impact that this disease has on someone's life. So when we think about treating diabetes, we know that it's a disease which is characterized by elevations in blood glucose levels. And looking at many of the various treatments that are currently on the market, we know that there's a broad range of treatments that have success in alleviating this hyperglycemia. And the reason a lot of patients will end up with starting with medications such as metformin, but over time will actually require treatment with multiple different drugs. And the reason this is successful in mitigating disease is the fact that many of these treatments have effects on multiple different tissues. So when we think about looking at new treatments for disease, it's important to understand on a tissue level what's happening to these tissues with diabetes. So today we're going to focus really on two of these tissues, the pancreas as well as in the gut. And when we look at these organs from the lens of what my lab does research is that we're really interested in understanding the question of cell identity. What makes a cell a cell? So we know that cells have normal function, and under normal homeostasis, they're able to do their job quite well. However, with disease, they become dysfunctional and are no longer able to do what they need to do. So our thought is if we understand how these cells become altered with disease, perhaps we can reverse them and restore function, restore their ability to function like they do during normal homeostasis. And when we look at this specifically through the lens of endocrine cells or the hormone producing cells within the tissues. And we do this because we know that endocrine cells specifically become dysfunctional when they're exposed to metabolic stress, such as diabetes as well as obesity. And they're no longer able to keep up with metabolic demand and lose their ability to function to produce and secrete hormones. And what has also been identified is that endocrine cells, a lot of times, although they can still be there with disease, what happens is they actually have an altered identity. So that's what really my lab is interested in, understanding how their identity becomes altered. So today I'm going to tell you about two different projects in my lab. First, where we look at the pancreatic beta cells that produce insulin. And then second, we'll look at some how gastric endocrine cells are actually altered by diabetes as well. So first, looking at the pancreatic beta cells. So when we look at diabetes from the view of the beta cells, we know that beta cells tend to be organized structurally in pancreatic islets, and we know that their job is to produce and secrete enough insulin to maintain proper glucose homeostasis. However, when they're exposed to metabolic stress, they become dysfunctional and they undergo beta cell failure. So when we look at this failure, it's multifactorial and quite heterogeneous. So we know that some beta cells have impaired function, so they're no longer able to secrete proper amounts of insulin. We know that other beta cells can actually undergo cell death. And then we have another population of beta cells that actually lose their identity. And importantly, when we try to understand what's happening, we can look at each of these things on a cellular level. So with impaired function, we can use markers to look at dysfunctional beta cells. We can look for apoptotic cells using markers such as cleave caspase 3 to assess cells that are dying. And then when we think of loss of cell identity, some of the features that we see in beta cells is one, they become multi-hormonal. So now they're not just expressing insulin, but other hormones such as glucagon. Some cells also de-differentiate or revert into this progenitor-like state. And then it's been identified in literature that we have the appearance of cells that are positive for the mesenchymal marker vimentin. So when you look here, this was a paper that actually identified that you have vimentin, shown here in the white immunohistochemistry, that co-localizes with both insulin, shown in the red, as well as with glucagon in the green. So although these vimentin cells, the appearance of these vimentin cells has been characterized, we're not really quite sure what they mean or what they represent in the context of the pancreas. So we hypothesize that actually these vimentin positive cells are a unique population of failing beta cells that appear with diabetes and obesity. So when we look at that, why is it interesting that these cells acquire vimentin positivity? So we know that vimentin is a marker of mesenchymal cells. And this is quite distinct from the beta cells shown here in blue that are endodermally derived or epithelial in nature. So beta cells are normally quite structurally organized. However, one thing that we know is we do in other contexts, we have characterized, particularly in cancer, that some epithelials can acquire mesenchymal-like characteristics. So here we hypothesize that perhaps some beta cells with diabetes actually undergo this epithelial to mesenchymal transition with disease. And in order to test this and look at this more closely, importantly, we can take advantage of what is known in the cancer field and the fact that this program tends to be driven by transcription factors, EMT transcription factors, which I show here on the bottom, such as slug and snail, which repress this epithelial identity, but then activate this mesenchymal identity and can be characterized by various markers, including upregulation of N-cadherin, as well as the mesenchymal markers alpha smooth muscle actin and, of course, vimentin. So we first wanted to see, are we actually getting activation of this EMT-like program with diabetes? So in order to look at this, we turned to, my lab uses a lot of different mouse models of diabetes. So here we turn to the genetic model of diabetes, the DB-DB, where there's a point mutation in the leptin receptor, which causes these mice to overeat and become obese and thus develop diabetes. And here our control is heterozygous for DB. And you'll see when we stain the mouse pancreas for vimentin, in our control animals, while we do see vimentin positivity throughout the islet, which is reacting with endothelial cells or blood vessel cells, what we were really excited to see, if you look at the red arrows in the DB-DB staining, we're actually getting acquiring vimentin positivity in cells that look more epithelial in nature. To really make sure that that's what these cells are, we then co-immunostain first for the endothelial marker, CD31 shown in green. And you'll see again at the red arrow, we see vimentin positive cells that are not co-localizing with this endothelial marker, demonstrating that they're not endothelial cells. But then if we co-stain now with the hormone insulin, shown in white, you'll see that we get nice positivity in vimentin positive cells that also co-stain for insulin in the red arrow, while we're still able to detect these endothelial cells, showing the red arrow. And importantly, we see this not just when we use vimentin as our mesenchymal marker, but we see the same thing when we use alpha smooth muscle actin, shown here in red. So when we co-immunostain at the yellow arrow, we see alpha smooth muscle actin positive cells that co-stain with insulin, again in white, but are negative for this endothelial marker CD31 in green. So it does look like we're getting activation of this mesenchymal-like program. But is it also driven by this EMT transcription factors? So in order to look at this, we actually can isolate islets from the mice. So here we have our control mice labeled here as WT or wild type, and islets isolated from our DB-DB mice, shown here in DB in the blue bars. What you can see is we get a striking increase in specifically gene expression for the EMT transcription factor snail, shown here on bar 2. And importantly, we see this increase not just in mRNA, but when we immunostain for snail protein using an antibody that recognizes both slug and snail in green, you'll see that in our control islets, here we outline the islets with this white dash. In our control mice, we're not seeing nuclei positive for slug snail. However, in our DB-DB mice, we have some islets where we actually get complete activation or increase in snail positivity, or almost complete, not complete. But then when you quantify these cells, approximately 30% of the cells become positive, of the beta cells become positive for slug snail in the DB-DB tissue. So it does look like we're getting increase in this EMT transcription factor, but is this actually driving vimentin positivity? So then we co-stained for both vimentin and slug snail. We're really excited to see that it looked like the same cells that became positive for slug snail also would become positive for vimentin, shown here by 2 examples. So this was really exciting and led us to really believe that we were getting activation of this program. And since this is just one mouse model of diabetes, we wanted to see whether this could be a broad program that was happening. So we co-stained tissue from a human pancreas, and full disclosure, the antibodies don't work quite as well in the human tissue. But I hope you can appreciate that we are getting activation of this program, cells that become positive for both alpha smooth muscle actin and snail that also co-stain for insulin in human beta cells, which we were really excited about. So we see activation in human. And importantly, we can also feed mice a high-fat diet to induce diabetes, and we see the same thing. So in mice that have normal chow, we do not see vimentin positive cells, but in the islets from mice that have been fed a high-fat diet, we see nice induction of vimentin positivity in cells, shown here by the white arrow, and these cells co-stain for the hormone insulin. So we really, this was really exciting because we're clearly seeing this program activated. And importantly, I had mentioned before that when we think of loss of cell identity, there are several different features that have been characterized. So we did, although I don't have time to show it today, we do know that these vimentin positive cells, the majority of them are also multi-hormonal in that they co-express glucagon. But then when we also look at dedifferentiation, here in a lot of this work was done in my former postdoc lab where we found a marker for dedifferentiated cells was ALDH1A3. So when you co-stain tissue, here we're looking actually at OB mice, but co-stain tissue for vimentin positivity and ALDH1A3. What was quite striking is the cells that became positive for vimentin were completely separate from the cells that became positive for dedifferentiation. So we do think it's a distinct program of loss of beta cell identity with diabetes. So with that, we see this program activating, but is it actually being initiated in the beta cells themselves? So in order to look at this, we actually turn to some lineage tracing. So we know that the pancreas is derived from endoderm. And when we look at the pancreatic precursor, we know that endocrine cells are actually derived from a population of endocrine progenitor cells that express the transcription factor neurogenin 3 or are positive for neurog3. And it's these progenitor cells that can then differentiate into various hormone producing cells within the pancreas, the predominant cell types being beta cells, alpha cells, delta cells, and PP cells. So we hypothesize that these vimentin positive mesenchymal cells are derived from beta cells. So if we mark these beta cells with the fluorescence reporter in the insulin positive cells, they should become fluorescent. Here we're using a reporter that turns to green. Whereas if these cells were derived from mesoderm, as most vimentin positive cells are, they would be in this insulin promoter, they would not be marked. So we did this using a fluorescence reporter, which is normally fluoresces red, but upon Cree recombination actually fluoresces green. So here you'll see when we cross these RTG mice with the rat insulin promoter driving Cree, you'll see that while most pancreas is red colored, so we see the exocrine cells in red surrounding the islet, as well as these endothelial cells within the islet remain red. However, the beta cells become marked green, shown by the picture below. So when we co-stain this tissue, now these again are DB mice, in addition to the Cree and the fluorescence reporter. So when we co-stain for both insulin and vimentin, we can actually now see whether these vimentin positive cells, what color they are. So when we zoom in, what we were really excited to see was the fact that these vimentin positive cells in the red arrow actually are green, meaning that they were derived from this rat insulin promoter cell, and also co-stain for insulin in white. So it does seem these cells are derived from insulin-producing cells. And now what this fluorescence reporter allows us to do is actually disperse islets and isolate just this green fluorescent beta cell population. And when we do that and look at beta cells from either wild-type mice or DB mice, we again confirm that we see this increase in vimentin mRNA. What this allowed us to do is then screen to see if we could find another driver of this program. We actually stumbled upon the transcription factor YAP1. So here you'll see that YAP1 mRNA is quite strikingly induced in DB islets or beta cells from DB mice. And this was really exciting to us because we know that YAP1 normally is not present in adult beta cells. So it is being induced. And it's being induced not just on the mRNA level, but we always also see induction of YAP1 protein, shown here by immunostaining in green and the white arrow in the islets from DB mice. And importantly, again, we see this in more than one model. So here we see YAP1 induction in mice that have been fed a high-fat diet, as well as YAP1 induction in islets isolated from patients with type 2 diabetes compared to healthy individuals. So with that, we wanted to test whether YAP1, the transcription factor, was actually driving this program. So we turned towards Min6, which is a mouse insulinoma beta cell line. Here we overexpressed YAP1 linked to a GFP protein. You'll see that YAP1, when it's overexpressed, as we can mark both by the GFP in green as well as YAP1 immunostaining in white, we see that about 80% of the cells now become positive for vimentin, indicating that YAP1 is possible to drive this vimentin gene expression. However, when we do the converse experiment and overexpress vimentin GFP, we see no cells that are positive for YAP1, meaning that vimentin is likely downstream of that. Now, this allows us to further look to see what's driving YAP1 induction in these cells. And it had been previously established in cancer literature that dysfunctional mitochondria can increase YAP1. So we wanted to look at this closely, and what we did is we used the fluorescent stained TMRE, which binds to healthy mitochondria. So when we separate cells with healthy mitochondria, shown here in the red bar, versus unhealthy mitochondria, shown in the white, we see that the cells with dysfunctional mitochondria have increased levels of both YAP1 as well as vimentin mRNA. And then when we directly induce mitochondrial dysfunction, here using antimycin A or on the mitochondrial decoupler, FCCP, when we treat DB islets, we're actually able to induce YAP1 when we trigger this mitochondrial dysfunction. So it does seem that mitochondrial dysfunction is driving this increase in YAP1. And although I don't have time to go into it today, we've further able to find that this is actually driven by reactive oxygen species. So we know, have a good idea of what's going on to increase YAP1, but are we able to actually reverse this process? So in order to do this, we took advantage of what we know about how YAP1 functions. So we know that YAP1 is a transcription factor, which translocates to the nucleus to increase transcription of genes related to EMT. However, it's also regulated downstream of hippo signaling, where it becomes phosphorylated to lead to its degradation. So we can activate degradation of YAP1 by the small molecule atriclin B. And when we treat DB islets with LAT-B here, shown, you'll see that in the green bars, the islets treated with LAT-B actually have less levels of both vimentin and alpha smooth muscle actin mRNA. We can also block YAP1 by preventing its transcriptional activity. And just to show you one of the experiments here is we're also able, when we block YAP1 transcription, we're also able to diminish snail mRNA levels. So we are able to reverse EMT-like activation in these cells. But does that actually have any relevance to the disease itself or onset of diabetes? So in order to look at this, we actually took advantage of some YAP1 fox mice, which we could cross to RIP-Cre to delete YAP1 specifically in beta cells. And here we're inducing diabetes by high-fat diet feeding. So you'll see here that both our control mice as well as our YAP knockout mice in purple on high-fat diet gain weight, although there are minor differences, nothing that's significant. But when you look at fed blood glucose levels, what we were really excited to see that although the control animals in blue do have elevated glucose levels the longer they're on the high-fat diet, our control mice lacking YAP, or our mice lacking YAP1, had very minimal changes to blood glucose levels, indicating that they were protected from developing diabetes. We're able to metabolically challenge these mice as well by doing a glucose tolerance test. And you'll see again our control mice have impaired glucose tolerance. However, our YAP1 knockout mice, in response to that same glucose challenge, are able to clear their blood glucose more quickly, showing that they are metabolically in a better state. And importantly, we can also look at a second model of diabetes. So when we cross these YAP1 knockout mice, here we're looking actually at female mice, crossed to DB/DB mice. You'll see that, again, we see minimal differences in body weight, although they both are quite obese. You'll see they're around 50 grams. But our blood glucose levels in our DB/DB mice lacking YAP1 are actually quite normal and comparative to lean control mice. And then when we again look at an IPGTT, they're quite protected and more sensitive to a glucose challenge, our knockout mice, as well as when we look at insulin levels. They have a lot better insulin response. And although I couldn't show you the data today, we've also done glucose-stimulated insulin secretion experiments in isolated islets and see that these knockout islets respond better and able to secrete insulin better. So with that, that's the first part. What I've shown you here is that we do see this EMT-like program activated in both human and mouse beta cells with diabetes. We know that this program is really characterized by the EMT transcription factor snail, as well as mesenchymal markers, both vimentin and alpha smooth muscle actin. We know that drivers of this program are both YAP1 induction as well as mitochondrial dysfunction. And importantly, when we block YAP1, we actually alleviate diabetes in mice. But as with everything, there's always more work to do. So with that, some of the things that we're looking at a little bit more is first, is activation of this mesenchymal-like program reversible in mice, as well as can we isolate EMT-like cells from human islets to study the mechanisms more in depth? And also, how does this program actually intersect with some of the other pathways of beta cell failure? So just to show you a little bit of what we're doing right now in the future is we actually noticed that one of the markers, mesenchymal markers, that was induced in DB islets was this N-cadherin. And we can actually block N-cadherin by using a blocking antibody. When we treat, here shown with islets, when we treat islets with this blocking antibody, we're actually able to both decrease both vimentin as well as snails. So this is something that we're looking at doing treatments in mice now. One other thing that I also wanted to mention, it's not the best picture, but we do have preliminary evidence that suggests that this EMT-like program might also be induced in type 1 diabetes, as well as with obesity. So with that, I'm now transitioning to the second part of my talk where we look at gastric endocrine cells. So shifting gears a lot, one thing that we know is obesity increases the risk of developing many different types of gastrointestinal diseases. And this occurs throughout the whole GI tract, such as within the colon, you're particularly susceptible, increased risk for colorectal cancer, as well as other parts such as gastritis in the stomach and GERD in the esophagus. However, the mechanisms driving, particularly on a cellular level, these diseases, it's quite unclear. Now, one thing that has been characterized is the fact that intestinal LGR5 stem cells actually do increase in mice that have been fed a high-fat diet. So it does seem like on a cellular level, we're altering the homeostasis of cells. However, it's very challenging to look at endocrine cells, at least in this experiment, because enteroendocrine cells in the gut only make about 1% of total cells in any population. But when we look at it, we know that they're important because the gut actually secretes more than 20 hormones that are essential in various processes, such as regulating metabolism. Now, so when we look at just kind of how high-fat diet affects the GI tract, I came across this really, I think, very interesting paper that looked at just gene expression changes within the whole regions of the GI tract. So you'll see, we see the stomach, the small intestine, such as the duodenum, jejunum, and ileum, as well as the large intestine within the colon. And here, although the specific changes aren't as important, we see gene expression is a lot more perturbed, shown increase in yellow and decrease in blue. We see overall more gene expression changes in this first bar, the stomach, compared to the rest of the GI tract. Now, this is OB mice, but we see the same thing when we look at other models of obesity, such as high-fat diet. So I wanted to delve into a little bit more of some of these changes that we see specifically within the stomach. So we know, just like in the pancreas, gastric endocrine cells are derived from a NurOG3 precursor. So these endocrine cells within the stomach can then be marked by the general marker chromogranin A. Within the stomach, one important endocrine cell, when they secrete hormone, one important one is the one that produces ghrelin, and ghrelin is a hormone that stimulates appetite. Now, as I just showed you before, we know that we see more mRNA changes in the stomach compared to the intestine of obese mice. And there has been papers which have looked at specific populations, and it seems like diabetes and obesity alters the function as well as the distribution of endocrine cells in the stomach, but the mechanisms are not very clear at all. So what we hypothesize is that perhaps obesity and diabetes, because it's affecting more than one endocrine cell population, perhaps it's actually acting on the endocrine progenitor cell, the NurOG3 positive cell within the stomach with disease. So in order to test this, we took advantage of the fact that we have a lot of fluorescent reporter mice to identify these cells. So we have a NurOG3 GFP reporter mouse, which here the NurOG3 endocrine progenitor cell will fluoresce green. So we can actually similarly feed mice a high-fat diet and then isolate just these GFP positive cells and look at changes of abundance. What we were really excited to see is we do see increased abundance in NurOG3 GFP positive cells in mice that have been fed a high-fat diet. And importantly, we also see this same increase when we look at a different model of obesity and diabetes, here mice that have been crossed to DB/DB. So we do see more endocrine progenitor cells, but how are they altering the differentiated cells? So here we lineage traced all the cells derived from NurOG3 cells using a NurOG3 CRE that has been crossed to another fluorescent promoter, here tomato, which becomes red upon CRE reactivation. When we do that and isolate these positive cells again by FACS, similar to what we saw with the NurOG3 cells, we also see an increase in lineage committed endocrine cells in DB/DB mice. And importantly, we don't just see this increase in fluorescently positive cells, but when we look at specific cell populations, here we're showing all endocrine cells using chromogranin A in green, as well as those that produce ghrelin in red. We see an increase in both of these cell populations in mice that have been fed a high-fat diet, which we were really excited about. So we're seeing more progenitor cells as well as more lineage-committed endocrine cells. Well, what's the reason for this? So we first, given the studies that had been done in the intestine, we first looked, since there's also an LGR5 stem cell in the antral stomach, we used reporter mice to look whether LGR5 could be driving these differences. But just to cut to the point, we saw no change within the stomach, although we did see the increase in the intestine. So this led us to believe that it wasn't the LGR5 stem cells driving this increase. So then we went and thought that perhaps it was actually something in the neurog3 cells themselves that was driving this increase with disease. So to look at this a little bit closer, we actually zoomed in on the transcription factor FOXO1, which we had developed a reporter mouse linked to Venus to isolate these cells. And again, using FACS, we can actually use endogenous fluorescence to isolate FOXO1 expressing cells from primary mouse stomach. When we do this, again, we can confirm that we're
Oh,
getting isolation.
I want to talk to you, but
Yeah.
I have to turn this down because I can't hear it. And it's not even here. It's not at Morehouse. It's just it's about what I'm doing, and I can always go look at it again another time. Except
Okay. Okay. Yeah, no, definitely. Okay. So we see this increase in FOXO1 expression, and we're actually seeing that neurog3 is also enriched in the cells that are expressing this FOXO1 transcription factor. So then when we look at neurog3 cells, we wanted to see how many co-express FOXO1, and we saw striking co-localization and enrichment of this transcription factor, FOXO1, in a large proportion of neurog3 progenitor cells. So when we then isolated these cells specifically, again, now we're isolating actually neurog3 GFP positive cells. Again, you'll see that it does seem to be this neurog3 population, which is enriched for FOXO1 under control conditions. But what we were really excited to see is we actually saw quite a striking increase in FOXO1 gene expression in the neurog3 GFP cells isolated from DB mice. So then we wanted to test what FOXO1 could be doing in neurog3 cells. So to do this, we took advantage of a mutant mouse, which is constitutively active for FOXO1, here shown FOXKR. When we make primary stomach cultures of these cells, here these are 2D cultures, we were actually seeing more ghrelin positive cells in the cultures made from these cells that are expressing active FOXO1. And importantly, we see this not just in primary stomach culture, but actually in the tissue itself. We see, particularly in the corpus shown here on the fourth bar, that we see more ghrelin producing cells in the tissue of mice with this active FOXO1 mutation. And as I had previously mentioned, ghrelin stimulates appetite. So confirming this, we actually saw an increased food intake in our FOXKR mice. So it seemed like FOXO1 is driving this differentiation program in neurog3 cells to stimulate ghrelin production. But how is obesity altering this program? So we screened a lot of different, particularly developmental pathways, but we stumbled upon the Notch signaling pathways. So here we're showing immunostating for HES1, which is a transcription factor activated downstream of Notch. You'll see in normal standard chow tissue of in the stomach of just wild type mice, you'll see that normally HES1 activity is restricted to the very bottom of the gland, shown here by the red arrow, which this is where we get active, is active in the stem cells. But when we look at the tissue using immunohistochemistry of mice that were fed a high-fat diet, what was quite striking is the fact that now we're getting HES-positive nuclei throughout the whole glandular epithelium of the stomach. So it was quite highly induced. So we wanted to test whether perhaps it was Notch signaling in neurog3 cells that was driving this. So again, we first isolated neurog3 cells from DB neurog3 GFP mice. and saw that these cells in DB mice had quite an increase in, yes, HES1 mRNA. And importantly, since we can also block Notch signaling by treatment of primary stomach cultures with a Notch inhibitor, here shown DBD, we can confirm that we're able to decrease HES1 activity. But then when we look specifically at neurog3 gene expression, we actually decrease neurog3 when we block Notch, indicating that Notch does seem to be important in driving this pathway. However, how is it affecting endocrine cells? So in order to look at this, we now did the converse experiment where we actually increase Notch signaling. Here we have mice that can overexpress a Notch intracellular cellular domain, or NICD, and we get increased Notch when we combine with Cre recombinase. So here we can treat primary stomach cultures with recombinant Cre. And we see that when we do that, the cultures with active Notch, shown here by the second bar with Cre, we actually see more chromogranin A positive cells. So it seems like Notch activation is increasing chromogranin A within the gastric primary cells. So this is just treatment and activation of Notch on a broad scale, but we wanted to make sure that we could actually track the specific cells that activated Notch to see whether they became endocrine cells. So in order to look at that, we added our fluorescence reporter to this and found that the cells that had Notch activated had increased gene expression of both chromogranin A, the general marker, as well as ghrelin. So it does seem that Notch is driving this increase in this differentiation into endocrine cells. So we see both Notch and FOXO1 having an important role in this process, but we still don't know which comes first, which is the chicken or the egg. So in order to test this, we created primary stomach cultures using our active FOXO1 or FOXKR mice and blocked Notch signaling using the DAPT. And what we saw here is we saw very little difference between any endocrine cell abundance, here ghrelin in red or chromogranin A in green. So this led us to the mechanism that we believe obesity triggers this expansion of neurog3 endocrine progenitor cells by activating Notch in these cells, and through FOXO1 drives the differentiation into ghrelin producing cells. So this was exciting, but again, bringing it back to now diabetes, does this have any role in actually driving some of the metabolic consequences? So what we did here is we again fed mice a high-fat diet. So our control mice fed a standard chow in blue, and our mice that were fed a high-fat diet in orange, you'll see that when we look and do the GTT or glucose tolerance test, we see impaired glucose tolerance. However, when we inject this Notch inhibitor to mice that have been fed a high-fat diet, we were very surprised to see that these mice behaved more closer to our standard chow controls. So they were quite more sensitive to a glucose challenge. When we looked at the stomach of these mice, we were really excited to see that now these mice where Notch had been blocked have less chromogranin A positive cells as well as less ghrelin positive cells. But we know this was just an inhibitor, a general inhibitor injecting into mice. So we wanted to get a little bit more specific on what was going on. So this is just a quick schematic of Notch signaling from one of my collaborators. And we know that Notch is activated between cells where you have a Notch ligand on one cell binding to a Notch receptor on another cell, which then allows cleavage of this Notch intracellular domain to increase gene expression of the target genes. So we wanted to look at various Notch ligands and came across the Notch ligand JAG1 that actually seemed to be quite strikingly upregulated, looking at immunofluorescence in the stomach tissue of mice fed a high-fat diet. So then if you look again at our NEROG3 GFP cells, while we see minimal change in other Notch ligands, so here I'm showing DLL1, which has been shown to be important in regulating stem cell homeostasis, we saw minimal change in mice, cells isolated from mice fed a high-fat diet. However, when we look at JAG1, we saw almost a doubling of JAG1 mRNA with high-fat diet. And then we're actually able, and this is preliminary, so we're still working on validating this once I get all of my mice transferred to Emory. But one thing that we were really excited to see is that it does look like when we knock out JAG1 in NEROG3 cells, here using this NEROG3 CRE, in mice fed a high-fat diet, they do seem to be a lot more sensitive to a glucose challenge. So we're really excited about that, and definitely following up with some changes on the cellular level as well now in this tissue. So with that, what I've shown you now for the second part is we do see an expansion of both gastric NEROG3 cells as well as lineage-committed endocrine cells with obesity. We see that this gastric Notch signaling is also increased with obesity. And we think this expansion is really driven in a FOXO1 and a Notch-dependent manner. And importantly, when we think about translating it to disease, we do see that when we block Notch signaling, we improve both glucose sensitivity as well as these changes in endocrine cell abundance. So some of the things that we're looking at now is what regulates this cell-specific increase of Notch in the NEROG3 cells, as well as how is FOXO1 promoting the expansion and differentiation of NEROG3 cells. So with that, just kind of to show you some things that we're working on with that first part is we're actually have been able to do both a bulk RNA-seq as well as a single cell RNA-seq on some of these cell populations with obesity. And I wanted to show you here that when we look at bulk RNA from NEROG3 cells, we see pathways involved in mitochondrial function. So oxidative phosphorylation as well as reactive oxygen species seem to be increased in NEROG3 cells from DB mice. Also, these other disease processes that show up in the pathway analysis are actually due to this increase in a lot of genes that are involved in mitochondria. And also another interesting thing is when we look at just the top genes in these cells from DB mice, is we actually see an increase in a lot of the gastric hormones, so gastrin, ghrelin, somatostatin, as well as the general endocrine marker chromogranin B. So this is something that we're really excited on and continuing to work on. So with that, now just bringing it back to really our focus on endocrine cell identity, and ultimately with the goal is can we reverse some of these changes that we see with disease in order to develop new therapies to treat particularly diabetes. And really our focus in the lab right now is both on the side of the gut, where we're looking at regional specific programs. Some of the projects I talked about are obesity-induced plasticity, as well as we also have another project where we're reprogramming gut NEROG3 cells into other cell populations. And then on the other side of my lab, we're really focused on the pancreas and this EMT-like beta cell failure. So how does it interact with other pathways of beta cell failure or in other contexts such as type 1 diabetes? We also have a very exciting collaboration in pancreatic cancer. So with that, I'm open to collaboration. So if you have any great ideas, I'd be happy to talk more. So with that, a lot of this work was an extension from my postdoc with Mimo at Chile. So I just wanted to thank all the members of the laboratory and the collaborators who maintain to be collaborators with me on this project. Of course, I want to thank my labs. So I started at Emory this past January. So this was our first lab picture with two of my undergrad students this summer. And then we also have some other undergrads, Matthew, Avi, as well as a PhD rotation student, Ariella. So excited about that. And I am hiring, so feel free to let me know if you know of anyone. Of course, I'd like to thank my funding, as well as I'm part of the Division of Digestive Diseases, and it's been a great environment to get off started with my lab on the right foot. And we're collaborating with Charles Cho on the pancreatic cancer project, as well as, of course, thank all the different centers, as well as the Global Diabetes Center for really welcoming me with open arms, and just thank you for your attention.